Hello, everybody. Welcome back. We are going to talk about lipids today. So, another type of macromolecule that we must understand and know the function, why do we need lipids in the body, etc. So, I have on my title slide here some examples of some lipid-based foods that you might consume. Over here, I have a picture of this pie chart where most of your lipids are triacylglycerols or glycerols, which are sources of fat and oils found in plants such as olives, corn, nuts, milk, and cheese. Tiny percentage of your fats are sterols, which are manufactured by the body, and 2% are found as phospholipids, which are around each cell encompassing the cell membranes. So let's get to it. Lipids are mostly made up of carbon and hydrogen atoms and some oxygen atoms as well. So we would consider lipids organic. They're very high in hydrogen to oxygen ratio, much more than 2-to-1. And they're typically made of two parts, a glycerol and a fatty acid. If you recall, fatty acids are some of the building blocks of lipids, and glycerols are as well. So let's go ahead and take a look at some examples of some lipids that we might find. The main one that I'm going to have you know are called triglycerides. The prefix tri means 3, so there's going to be some type of 3 component in there. So 1 glycerol molecule in a triglyceride, and you bet it, 3 fatty acid molecules in a triglyceride. Triglycerides are known for our dietary use, so when consumption, energy source, and insulation. Phospholipids are going to be those surrounding or encompassing cell membranes. Flexible fences, I like to call them. They're a cell membrane barrier. They're made up of glycerol molecules, phosphate groups, and 2 fatty acid molecules. If you recall, phosphate group is one of our functional groups that we need to know. Next are waxes. Waxes are going to be long fatty acid chains and long alcohol chains. They are waterproof and they provide protective coating. Steroids are the last type of lipid that I want you to know that have 4 fused carbon rings with functional groups attached. And they are encompassing hormones, which are our chemical messengers in the body that travel the blood, like testosterone. Cholesterol, sometimes you find embedded in our cell membranes. And estrogen is another type of hormone. So here's our functional group chart that you're going to eventually need to have down. You are going to be looking for some of these functional groups in various macromolecules. We've already really honed in on the hydroxyl groups. And we did try to spot in amino acids, carboxyls, and amino groups. Phosphates, I already mentioned here that you are going to need to be able to recognize. Phosphates are going to be in phospholipids. And let's go ahead and take a look at the structure of glycerol, which is a component of lipids. So to make a lipid, we need to have 2 building blocks. The first part is a glycerol. So this is a glycerol. It serves as the backbone of the lipid. Okay, so if you notice from our previous functional group chart, the glycerol has some hydroxyl groups here. 3 hydroxyl groups. Okay, so in order to make a lipid, we need the glycerol and we also need the fatty acids. So fatty acids are long hydrocarbon chains, so hydrocarbon, hydrogen and carbons together, and they bond with a carboxylic group. Okay, so that is going to be a group that's also found in our functional groups table. Now, these carbon chains with those hydrogens attached are very long chains. And this one happens to be a straight kind. So you see how I can just draw my line that's very straight? We call those saturated fats or saturated fatty acids. Let's take a look at some of these examples. So here is the straight chain I just mentioned. That one is called a saturated fat. I'm going to make sure that matches the color of our line because typically saturated fats are considered bad fats. So red means bad in our case here. So all single bonds in the carbon chain, you have a maximum possible number of hydrogens. They're typically very straight and they're generally considered bad for you. Now saturated fats, one of the conditions that they're in is that it is a solid at room temperature. So examples of this is like butter, ghee, animal fats, all of those are going to be examples of saturated fats. On the flip side, our unsaturated fats are going to be one or more double bonds in the carbon chain. So you can see here this like kink in the chain. There could be more hydrogens. They're not straight and they're considered better for you, probably because since they have that little kink in them, they don't stack as well as the saturated fats in the arteries and veins. So you see a little bit more blood flow with unsaturated fats. They are also deemed liquid at room temperature. Okay, these are going to be your plant-based fats and oils. So olive oil, veggie oil, etc. are going to be more unsaturated than saturated. So how do we make these lipids? How do we form the bonds between the glycerol, the backbone of the fat, and the fatty acids to make our macromolecule of the lipids? Well, we need to use our same good old process of dehydration synthesis. Dehydration, reminder, is to remove water. And then synthesis means to put monomers together. So it does not have to just be monosaccharides. They can be monomers of a lipid too. So monomers of a lipid is your glycerol and your 3 fatty acids. So take a look. A triglyceride is an example of a lipid. It's our number 1 in our list. And as you can see here, we have our glycerol backbone and our 3 fatty acids. So how do they link together? Well, we have our hydroxide here, our hydroxyl group, and it's going to link with the hydrogen. The hydroxyl group and the hydrogen on the other side, on the fatty acid, is going to be removed so we can make the bond between the oxygen here and our carbon here. So same way, here and here and here and here, I'm going to lose 3 waters. In the process, I'm going to make the 3 bonds. So how many water molecules were removed to create this triglyceride? Well, I see here 1, 2, 3. So 3 waters. It's not 1 less than the number of bonds in this case because I need to lose those 3 waters to make the 3 bonds for the lipids. So 3 waters removed to create the 3 bonds in the triglyceride. Now I like to make this look like a flag. Kind of looks like a flag to me. We see like this part, okay, the glycerol, and then we have the 3 fatty acids. And it looks like one of them is unsaturated because it has that slight kink to it. See? But it kind of looks like an American flag to me. So if you see that on our macromolecule chart, that's probably why that's what this is referring to. So in order to make and break a triglyceride, I need to either add water. If I add water, then I am going to break it apart. We call that hydrolysis here. And if I want to remove water, that's when I want to put them together here. So if I remove waters, then I can make these 3 bonds. Okay, so here is a video we can go ahead and watch now. I can link it as well, in our page. But obviously I'm recording this, so I'm just going to bypass this. Okay, so examples of lipids. We have meat fat, so fat like bacon grease or whatnot. oil, waxes, butter, grease, mayo. So all of those are examples of examples of lipids, and you should be able to classify if they're saturated or unsaturated given a list of examples. So here is a typical nutritional label, and what you're going to do is you're going to circle where the lipids would be located on this food label. So as you can see here, I'm looking for the fats. Okay, so they do have different types of fats, but this is where the fats are. The lipids are here. Okay, and then you can, continue this little fun game and do the carbohydrates as well. Here's the examples. Phospholipids, we have our phosphate group in there. Okay, phospholipids, these weird looking things, they look like jellyfish or something, they make up the cell membrane. So if I go ahead and show you what that looks like, lots of them put together makes up that fluid mosaic flexible fence cell membrane around each animal cell. And plant cells have it too, but plant cells also have an extra cell wall. Steroids, here's what steroids look like. Those are going to be those special classification of hormones. And waxes look like this. You can say like your earwax or whatever. All right, so the function of lipids, why do we have these things? Well, we need to have them because they are used for storing long-term energy. So your sugars sometimes convert to fats over a long period of time, or when you have an excess of carbs that you don't use, then they do convert to fats. They're used for insulation, so that, like not just us, but, other animals use lipids as insulation for maybe the winter, because they got to be outside. For us, we use them as cushion support for our organs, and they're also an important part of our cell membranes. So how do we test for lipids? This is going to be the fun part. Okay, so there's two types of tests we can test for lipids. The first test is solubility, and the second is the brown paper test. So the solubility test is a very strange test because basically if you are positive for solubility, that means that you are able to mix with water. So let's write that down. Soluble means mixing ability with water. Unfortunately, the problem with that though is that it's not that easy. If you are able to have a substance that is mixing with water, well, that means that substance is not lipids or oils, because oil and water don't mix. So as you can see here, this is a layer of oil and water. Okay, so they are not soluble. That means they are not mixing together. So write that down, not soluble. Well, if they are not soluble, if the solution is insoluble, we call it, or not soluble, that means it's positive for lipids, because lipids are present and you can see a layer. So you should say layer present. If there is a layer present that you see on top of the water, you know that there are lipids present, okay? That means that it is negative for solubility. So positive for lipids, negative for solubility, because it has there's no mixing ability with water, insoluble. So positive for lipids means negative for solubility. Okay, and that's what this information says. It says lipids do not mix in water, so anything that has the ability to dissolve in water is positive for solubility and therefore negative for lipids. That is very confusing for lots of children, and I apologize for that. But you're just going to have to know it. Know the relationship there. This is because lipids are hydrophobic. They have nonpolar properties, and they do not mix with polar substances. So a negative for lipids is here. Negative for lipids, you can see that there are no layers. So no layers present. That means that is negative for lipids and positive for solubility on this glass. All right. Next, brown paper bag test, or brown paper test. Our Fremd paper towels are perfect for this because they are terrible, but they're really good for the oil test. So or lipid test, excuse me. So what you do is if you put the unknown substance on a brown paper bag or a brown paper towel, and the bag dries very well over time, the substance is considered a non-lipid. So if you take a look at the picture, the right side is dried up and you can't really see, it's not translucent, it's not see-through. However, if there are lipids present, the bag will still look wet and light will be able to shine through. So lipids are present. So this one's kind of fairly easy, it's either present or not. Versus the other one, the solubility test, it's like the opposite rule. Okay, so hopefully you will be able to see your teacher do this test at some point in your time at school. So you can see if it's, positive for lipids on the brown paper towel or brown paper bag test. So here's where you pause and check for your understanding. Name 2 major similarities between carbs and lipids, 1 structure and then 1 function, and then 2 major differences between carbs and lipids, 1 structure, 1 function. And pause there, make sure you fill that out. And then we have our last part is that we need to fill out our macromolecule chart. So this should be posted in Schoology somewhere for you to fill out. Basically, what you need to do is you need to take time to fill out this old information, which is the carbohydrate section. The Lego pieces are just there to remind you that they are building blocks, just like Lego pieces are. We also filled out the lipids one here from today. So you should have the carbohydrates section, which is this section up here, and this section here. So what you're doing is you're filling this out best of your ability. Basically, in this first, you're going to write down the chemical formula for a monosaccharide and the picture of what it looks like. Disaccharide, chemical formula, picture, and then polysaccharide, maybe just a picture of a typical polysaccharide, with maybe the hexagon rings or whatnot. Synonyms, test to identify. So that's going to be the test to identify, like in lab, how are you going to identify the 3 types of carbohydrates? Here are some examples we talked about in class. And how do you recognize this in the body, or what is it used for in the body? And same for the lipid section. Do all of those things. Okay, I have a line between for the test to identify, so you can do the solubility test on one side of the box and the brown paper on the other. Okay, please fill this out to the best of your ability, and we're going to circle back soon. Thank you for watching. Bye.